This is a recording for my 112 students in the synchronous class today, which is March 24th, 2021. We came across this term, the Hebbian principle, which you can see here, the Hebbian principle. I'm assuming that's how you pronounce it. I um, I had not heard that before, so I wanted to make sure I looked it up and explained it to everyone. So what I did is I copied the information from our study plan on Moodle that goes into the content on the Hebbian principle. It involves two things: long-term potentiation and long-term depression. It's really interesting. So all of this is actually related to a concept called synaptic plasticity. Let me make it so I can make notes. Okay, so if I want to draw, I don't want to draw in red. Let's see. What about blue? Okay. All right, so this way I can draw on here. Um, so synapses are not static structures. Now remember what a synapse is. A synapse is, I can scroll down and show you, a synapse is what we see three synapses here. The synapse is where one axon terminal like this is an axon terminal, um, synapses or comes very close and interacts with either another neuron, or it can be a muscle, or it can be a gland, but it's some target. Okay, we're going to call this, in this case, it is a neuron, and the one on top is called the presynaptic axon terminal or presynaptic neuron because it comes before the synapse and it's the one that's going to carry the signal to the other neuron called the postsynaptic terminal or postsynaptic neuron. This is the synapse in here. This gap is your synapse. So when we say that synapses are not plastic, not plastic, synapses are not static. That just means that, I mean, think of it this way because I didn't realize this, but when I read about it, this actually can be related to drug addiction because there are pharmaceutical drugs that have effects on our nervous system. So obviously, there's going to also be street drugs that do the same thing. You know, um, I mean, just two, like probably the two most addictive ones that we hear about are opiates and cocaine type products. So, amphetamine type products and opiates. So, which there's more and more and more different types of opiates. But the point is, okay, you take the drug or medication one time and it causes a certain effect and then you develop a tolerance over time. This synaptic plasticity can help explain that whole, process. So synapses are not static; they can be weakened or strengthened. Now we did learn that an action potential is all or none, but if you want to get to an action potential, you have to have enough of your um, depolarization, excitatory potentials to add up to threshold at the axon, at the beginning of the axon called the axon hillock. So you have to have, okay, I'm doing a very bad job of explaining this. If this is your cell body of your neuron, and these are your dendrites, they're going to receive signals. Okay, this is going to be your axon. All right, I'm going to change colors. What you want to get to is right here. I tried to make that green. Let me try this again. I'll have to choose it. Okay, there we go. All right. All right, so right here at the beginning of the axon is called the axon hillock. Now, if at this axon hillock you get depolarization to threshold, then you will have an action potential and it will travel all the way down the axon to the axon terminals. So an action potential is all or none. You either have one or you don't. But the excitatory and inhibitory potentials that occur either along the dendrites or along the cell body, these potentials can either be, remember, EPSP or IPSP, stands for excitatory presynaptic potential or inhibitory presynaptic potential. So excitatory means that it leads to an action potential, but the excitatory potentials have to be strong enough that they allow depolarization to reach threshold at the axon hillock. So you can have depolarization, say at this point here, and it can spread out a little bit, maybe this far, but it weakens as it spreads out. And then eventually, I mean, it's not gonna, it hasn't reached the axon hillock. So you have to have another one. Maybe this one's a little stronger. And it has to add up. These have to add together to where they actually reach the axon hillock and cause a depolarization from -70, which is resting, the resting potential, to around -55. So they're going to have to allow for sodium ions to come into the plasma membrane and change that potential, change that voltage from -70 to -55. That's the threshold value in order for an action potential to take place. So what we're talking about are these excitatory and inhibitory presynaptic potentials. And so they can be weakened or strengthened. The point is they can be weakened or strengthened, but action potentials cannot. Action potentials are all or none, but presynaptic potentials can be weakened or strengthened. And here's how this kind of works. All right, one way is long-term potentiation, and this is what happens when you get a tolerance for a certain drug or tolerance, for medication or a street drug. All right, this is a persistent strengthening of a synaptic connection. The Hebbian principle says that cells that fire together wire together. Cells that fire together wire together. All right. So what does that mean? And then it goes on to tell you that there's various mechanisms that most of them are not really fully understood. But here is one known mechanism, and it involves a glutamate receptor called NMDA. So here is glutamate is your neurotransmitter, and glutamate is released into the synapse and it binds with an MDA receptor, and then it leads to calcium coming into the postsynaptic axon terminal, and those calcium ions are going to lead to an action potential. They're gonna, this might be like a dendrite, and if this is going to lead to, I'm sorry, this is going to lead to either an EPSP or an IPSP. And this could actually be an axon terminal, because sometimes axons meet up with other axons. There are situations like that, but more than likely this one right here is like the end of a dendrite, or it could also be part of the cell body. But anyway, the calcium ions coming in lead to excitatory potential. And if that potential spreads to the axon hillock, then you're going to have an action potential. okay, which is the goal. So what happens is when you get enough stimulation on the presynaptic neuron, when you get enough stimulation here, so either stronger potentials or more potentials here, that's going to lead to this magnesium, which was bound to this MDA receptor, is going to be kicked off, okay? And then it's going to lead to more calcium coming in than normal, like even more calcium than you would normally expect. And the calcium is going to lead to these AMPA receptors. And you can actually, the AMPA receptors can actually more can be added to the plasma membrane of the postsynaptic neuron. So the more AMPA receptors that you have, the more chance, the more stronger of a signal that you're going to get in your postsynaptic terminal. So it says these receptors, and it's talking about the MDA, these receptors are normally blocked by magnesium ions, all right? But when the postsynaptic neuron is depolarized by multiple presynaptic inputs in quick succession, so either you get a whole lot of different excitatory potentials or from different neurons, or you get a bunch, right, a whole bunch in a row from one neuron. It forces the magnesium ions out, and that allows calcium to come in. Okay, so that's what we're seeing happen in the picture on the left. All right, when the calcium comes in, they initiate a signaling cascade that causes AMPA receptors to be inserted into the postsynaptic membrane. And AMPA receptors allow positive ions to enter the cell, and positive ions are going to lead to an excitatory presynaptic potential. So the main thing to understand is the addition of these AMPA receptors. The more AMPA receptors you have, the stronger your signal is going to be. Okay? So the insertion of additional AMPA receptors strengthens the synapse. And that's what long-term potentiation is. It's the strengthening of the synapse. And that means the postsynaptic neuron is more likely to fire in response to presynaptic neurotransmitter release. And that neurotransmitter was glutamate. And then the last sentence is what refers to tolerance. Some drugs of abuse co-opt the LTP pathway. And this can lead to addiction and tolerance. And long-term depression is the exact reverse. It's just a weakening of the synaptic connection, and that instead of inserting additional AMPA receptors, it is removing AMPA receptors. And if you remove them, you're not going to get enough excitatory potentials to lead to an action potential in your postsynaptic neuron. So that is long-term depression, is removing the AMPA receptors, whereas long-term potentiation is inserting additional AMPA receptors. So that is the, I can't even remember the word now. Whole reason I did this, the Hebbian principle, cells that fire together wire together. So it's just the more excitatory potentials that you get, the stronger, the more likely you're going to get an action potential. And the wire is the axon that the action potential travels down. So that was real fast, but hopefully it helped you a little bit to understand the long-term potentiation and long-term depression.